No, they didn't isolate the virus. That's the issue. That's the issue.
Well, you isolate a virus by
Clearly, logic and common sense dictate that if you have not isolated the thing you are looking for, there is no possible way to determine what the thing is made of or what it alone does.
You may be interested to know that the prize for providing the proof of isolation and genetic content for SARS-CoV-2 has jackpotted up to €1 million.
We make it clear we're not just asking for something maybe where you yourself isolated. We're saying any record of it being isolated anywhere in the world by anyone, and they have nothing.
Yet all of the listed institutions indicated the same, that they had searched their records and located none describing legitimate isolation of any SARS-CoV-2 patient sample performed by anyone, anywhere on the planet, ever. And if the virus actually existed, should this information not be at their fingertips?
This is the entire gig right here. If these are not pathogens, then why are we putting masks on? And why are we social distancing?
I come from a scientific and medical background, so I'm able to read the literature and understand what things are what. And so I started looking at the articles where they say that they've discovered a novel coronavirus. And what I uncovered is that they put forth this experimental criteria that proves the existence of a virus. But when you take a closer look, it's actually the experiment itself that gives them the proof they need, even if you don't even have any source of a virus in the experiment at all. And this was actually carried out recently by Dr. Stefan Lanka in Germany, who showed that he got the same proof of a virus by doing the experiment without any source of a virus whatsoever.
The dying of cells in the test tube we can create in the same manner without infected material. And then they're calling their dying cell tissues an isolate, which then they offer on the market for €2,000. And then they say, and in here, and this is the virus. And from that we can create a vaccine now.
My co-author, Torsten Engelbrecht, made a point of contacting those authors who had claimed to have first identified or isolated the purported virus to see if they had purified samples. We published our findings in the recently released updated version of Virus Mania, and as many of you will know, none of the study authors claimed to have obtained the virus in purified form.
The most important paper written on the COVID-19 situation by Christian Drosten and others on where he got the genome or where he got the PCR segments that they're using in their tests. And he says, and I'm quoting here, "We aim to develop and deploy robust diagnostic methodology for use in public health laboratory settings without having virus material available." Now, I don't know how much clearer they can make it that they never actually had possession of a virus. They never had possession of a genome.
A CDC document titled Severe Acute Respiratory Syndrome Coronavirus 2 from Patient with Coronavirus Disease, United States, describes the process of what they call isolation of the COVID virus. The process starts with a swab taken from a male Chinese patient in Washington, D.C. in January of 2020.
They did this in a way where they had basically fragments of genetic material from whatever would be in someone's lungs who was sick, which would be a lot of different things. And there turns out that there's like 56 million individual little fragments of RNA, and they sequence those, and we don't know where any of those individual fragments are from because we didn't get it from a virus.
After acquiring the sample, substances were added to the mix. Among the substances added to the unpurified sample were vero cells, which are kidney cells from a green monkey. They also added human liver cancer cells, human embryonic kidney cells, human lung cancer cells, and fetal bovine serum. The mixture was then put into an incubator and later tested for cell damage. This cell damage they call a cytopathic effect and is assumed to be caused by the presence of a virus. It turns out that the only cells which were damaged were the vero cells, or the monkey cells. None of the human cell lines showed the cytopathic effects. In other words, only monkey kidney cells grew the virus. But as Dr. Lonka had discovered, no swab sample was required to produce the same cytopathic effects, just this witch's brew of substances added in the CDC's experiment. Next, they took samples from the damaged cell culture, enough, they say, to span the genome of the virus. But the genome at this point had not been mapped, so how did they know its length? Enter GenBank, a reference library of all known viral genomes.
So they pull out these sequences, and then of course, they're not a continuous sequence. So how do you piece them together? So basically a computer runs for several days trying to piece these things together in some theoretical framework that fits the parameters of whoever set up the operation. So they say, oh, well, it's going to have a spike protein and it's going to be at this point. So then the computer fills in sequences, who knows where they're from, that were from, somebody's snot. And then when there's missing gaps, the computer just makes it up. Like they may pull it from something in a database from another virus, or just generate it based on a pattern recognition algorithm, right? So essentially, it's just a theoretical model, and sometimes it's even referred to in the literature as an in silico model, right? Meaning that it's made in a computer on circuitry. It's not a real genome of a real organism. And anyone that, purports it to be, they should just simply look at the methods. And this is roughly called next generation sequencing.
When the computers had rendered their results, the data was, quote, interpreted manually, or more commonly using qualitative software by suitably trained interpreters. In other words, a panel and a computer tweak the final results. Let's recap the CDC's virus isolation procedure. They start with a swab from a person with symptoms. Then they add several toxic substances. Then they observed that monkey cells became damaged. They assume the damage was caused by not just a virus, but a human virus. Then they choose a virus from GenBank that they think it might be similar to. Then they run several computer algorithms to make the data fit that model. A panel of experts tweaks the final result to publish what will come to be known as the SARS-CoV-2 Complete Genome.
I started submitting Freedom of Information requests to Health Canada and other Canadian institutions to see if they had any evidence of real SARS-CoV-2 isolation. My requests were specifically worded to weed out the papers that had claimed to have isolated, but in fact performed what I call fraudulent monkey business. So far, the following Canadian institutions have responded: Health Canada, National Research Council of Canada, the Vaccine and Infectious Disease Organization - International Vaccine Center at the University of Saskatchewan, which had claimed to have isolated the virus, McGill University, the region of Peel, where I used to live, the City of Toronto, University of Toronto, Sunnybrook Health Sciences Centre, McMaster University, and Mount Sinai Hospital in Toronto. Note that researchers from the last 4 of those institutions had already publicly claimed to have jointly isolated the virus, and at least 2 of them are involved in COVID-19 vaccine development.
Now, if we go back to our dictionary definition of isolate, which is to obtain a substance or microorganism in an uncombined or pure state, we can see that people are confused, or at least talking about very different things, because nobody has produced an uncombined or pure state sample. Other investigators were also aware of the inconsistent ways in which isolation was being used. Here in New Zealand, the media reported in mid-2020 that Professor Quinone Matteau of Otago University was able to grow the SARS-CoV-2 virus and isolate its RNA, its genetic material. However, Canadian researcher Christine Massey and New Zealander Michael S. have been submitting Freedom of Information requests to health institutions around the world. When they made an Official Information Act request to Otago University, they were advised that the university in fact had no records of any study describing the actual purification of the alleged virus. Okay, it's time to get some more lessons from Professor Racaniello to see if a representative from the orthodoxy can tidy up their mess. He says, an isolate is a virus that we have isolated from an infected host and we have propagated that in culture. You put that sample in cells and culture, which is how we grow viruses. And you grow virus and now you have an isolate. Alright, I think we might have hit some major problems here. He's implying that someone spits in a cup and if you mix it with a cell culture, hey presto, we've isolated a virus. That doesn't sound like isolation at all because we're dealing with samples and culture mediums that contain all kinds of substances without any mention of purification. With regards to this, in Virus Mania, we quote Nobel laureate virologist Françoise Barré-Sinoussi as saying, you have to purify the virus from all this mess. Professor Racaniello goes on to say that most of the time we take this nasopharyngeal swab in the solution, we just do the genome sequence and we don't actually have a physical isolate virus, and that's very important. Okay, so that's a big concession. He admits many of these so-called isolates are simply detecting genetic sequences from crude clinical specimens. Unfortunately, Professor Racaniello's discipline for terms really seems to fall apart when he goes on to say this is a phylogenetic tree of 4,000 or so genome sequences of SARS-CoV-2 isolates. But then he goes on to recover from this slip and state that the dots are individual genome sequences which may or may not have an isolate associated with them. But can you see the major problem here? Detecting nucleic acid sequences does not equate to proof of a virus. In fact, the professor said so himself in his 2017 blog. Many laboratories choose to assay the presence of viral genomes by PCR. This is an acceptable technique as long as the limitations are understood. It detects nucleic acids, not infectious virus. However, even with the professor's stricter definition of isolate, that of a culture mixture, he's still not referring to a purified specimen.
The central question is, how does any scientist, any virologist, prove that a, quote, new virus is the cause of any illness? The answer is surprisingly clear and straightforward. It also mimics how any normal human being proves the causation of anything. The first thing one does is to find a number of people who seem to suffer from the illness in question. This could be COVID-19, AIDS, Hep C, or any other illness. Then, using standard and common virological isolation and purification techniques that have been in common use in every virology lab for decades. One would isolate the virus from every other substance in the blood, sputum, or tissues of the affected person or animal. Using electron microscopy, one then shows the world the pure isolated particles called viruses from each of these people. All of the viruses should look identical. The next step would be to analyze the genetic material of these identical particles using commonly available computerized genetic sequencing tools, which have also been available for decades. These sequences should also be identical from one particle to the next. Finally, these isolated, purified, photographed, and sequenced viruses would then be introduced onto test animals along with rigorous and appropriate controls to see whether the test animals develop the identical illness that the original subjects had. Then and only then, once these steps have been performed, can we say with confidence that this new virus is the likely cause of this new illness. During this past year, I have spent countless hours looking for evidence that these simple steps were done for the illness called COVID-19 and that the virus called SARS-CoV-2 was properly isolated and characterized. To my shock and surprise, neither I nor anyone else I know looking into this issue has yet to find a single published study that even attempted to perform these simple and doable experiments. As shocking as this may be to hear, I now ask that if you dispute this, please make available the paper or papers that successfully did these simple experiments.
So have particles that can only be seen on an electron microscope, been isolated in any context? And I would say yes. So they have found particles called bacteriophages that live in bacteria cultures in a pure culture in laboratory conditions. And also they have found particles of that size in other lower organisms like sea algae. And I think they call those giant viruses, actually. And those particles are quite interesting. And I would encourage you, I wish I had thought of this before, to find some photographs of the microscopic images of these particles, because they have very, very distinctive geometric forms. They don't just look like a round blob like the pictures of the alleged viruses that cause disease.
We also have a communication from the U.S. National Institute of Allergy and Infectious Diseases, which is Anthony Fauci's institute, indicating that the electron microscope images that they've published did not involve isolation of any virus.
I'll read you what Pfizer says about their mRNA vaccine. Quote, "The DNA template used does not come directly from an isolated virus from an infected person." Here's another quote. "The DNA template, SARS-CoV-2, gene bank, etc., was generated via a combination of gene synthesis and recombinant DNA technology." So if this is a hoax that Pfizer doesn't have the virus as their template, then apparently Pfizer is in on the hoax.
A casual reader, like a typical doctor who would, see articles and curated by some organization like WebMD, they would just see the titles of the article and they would think that the virus really was separated and isolated according to the definition of the word isolate that they know. However, that's been completely obfuscated in these papers by coming up with this false definition of isolation, which really means to grow a toxic cell culture and then show cell damage.
We have taken a major step for freedom by coming here today. We need to continue our efforts. We need to be prepared for sacrifice in the short term, but we are strong. We can endure. Let us make a commitment right here today that we will continue to act as the beautiful, amazing, free beings of light that we truly are. We will stand up to tyranny at every step. We will not comply. We will not comply.